This is the 12th video in the Fundamentals of Biology, and today we're talking about gene expression. So first, let's talk about the central dogma of biology. So the first step is transcription, and this is where DNA will have to become RNA. Because DNA cannot leave the nucleus, it must undergo transcription in order to generate mRNA code. The next step is translation, and the RNA sequence will undergo translation at the ribosomes of the cell to become a protein. So you can see in this first diagram, after the DNA is transcribed, it will leave into the ribosomes in the cytosol of the cell in order to code for proteins. The first step of transcription is called initiation. And a DNA sequence called the promoter will signal the start point. And this is called the Tata box because the first four nucleotides are TATA. Next, transcription factors are proteins that will help RNA polymerase II bind onto the promoter. And this final product is an initiation complex, which will begin transcription. Next, elongation will occur. In elongation, RNA polymerases will add nucleotides to the 3' end by using the 3' to 5' DNA template strand. So this bottom strand will be the template strand. However, the strand that is not being used is called the coding strand because the nucleotide bases are the same, except uracil will be present instead of thymine in the RNA transcript. The final step of transcription is termination as well as modification. So termination, after the transcription unit on DNA is done coding for the mRNA, a terminator, which is a DNA sequence found in bacteria, will cause the RNA to be released. After that, RNA has to be modified. The 5' end will receive a 5' cap. The 3' end will receive a poly A tail, which means it has a lot of adenines within the nucleotides. And the non-coding regions called introns will be removed through something called RNA splicing. And this is done through spliceosomes, which recognize these sites. The coding region called exons will be expressed. Finally, ribozymes are catalytic RNA molecules that will splice the RNA as well. In order to get from RNA to protein, we undergo translation. Here are some basic terminology that we need to be familiar with. A codon is a sequence of 3 nucleotides that will code for a certain amino acid. So in this picture, the first 3 letters is a codon, and it will code for a certain amino acid. mRNA, also known as messenger RNA, is the RNA strand that DNA transcribed into. Ribosomal RNA, or rRNA, will create proteins and contain a large and small subunit, and also an A, P, E sites for tRNA. tRNA, or transfer RNA, transfers an amino acid to a polypeptide chain using an anticodon, and an anticodon is 3 nucleotides which are complementary to a codon. The first step of translation is also initiation. So first, you will have a start codon, and this will be signified through AUG. Next, the small rRNA subunit will bind to the mRNA and the corresponding tRNA. In the third step, initiation factors, which are proteins, will bring in the large subunit to make a translation initiation complex. So this final picture will show a large subunit and a small subunit with a tRNA binding to the start codon. The next step is elongation. This will occur from 5' to 3'. The tRNA with the corresponding anticodon will attach at the A site. Next, a protein sequence at the P site will form a peptide bond with the amino acid at the A site. So in this picture, this polypeptide chain will be attaching onto the amino acid at the A site. Then the tRNA at the P site will exit through the E site, and the A site tRNA will move to the P site. So these tRNAs will shift over, and the one that was previously there will exit. The final step of translation is termination, and at the A site, it will accept a protein called a release factor, and instead of adding an amino acid, water will be added, and this will release the polypeptide, and the rRNA will disassemble into ribosomal subunits. Now let's talk about mutations. Mutations are changes in the genetic information that may cause the wrong protein to be produced, and mutations are caused by mutagens, like carcinogens, which are chemicals that cause cancer. A point mutation is when one nucleotide is changed. There are several types of point mutation. Substitution is when one nucleotide will be replaced with another. There are also several types within substitution. A silent mutation will have no effect on the amino acid. This is because several codons can code for one amino acid. Missense mutations code for the wrong amino acid. Nonsense mutation will create a stop codon instead of the intended amino acid. Insertion and deletion are also mutations, and this can cause a change in something called the reading frame that the codons are read in, and this can cause frame shift mutations. So previously, the codon pair would go CAT, TCA. However, in frame shift mutation, it would read ATT, leaving the C at the beginning. As a result, every single amino acid along this chain will be different to the original one. Thanks for watching this video. This lesson covered transcription, translation, and mutations.